Megazyme provides an extensive range of assay kits for use in various assay formats, including autoanalyzer, microplate and manual spectrophotometer. This acetaldehyde assay kit can be used to accurately measure acetaldehyde in various sample types throughout industries such as food and beverages. This video tutorial will demonstrate the use of the assay kit with a wine sample using the manual spectrophotometer format. It is important to measure acetaldehyde in wine as it can indicate oxidative issues during the wine making process. At low levels, it imparts a pleasant fruity aroma to wine, but is pungent at higher levels. This kit contains sufficient reagents for either 50 or 100 manual assays and is supplied with a detailed data booklet. The principle of the enzymatic reaction involved in the measurement of acetaldehyde is shown in this figure. In the presence of NAD, aldehyde dehydrogenase converts acetaldehyde into acetic acid and NADH. The amount of NADH formed is stoichiometric with the amount of acetaldehyde. It is the NADH which is measured by the increase in absorbance at 340 nanometers. Prior to sample analysis, the kit components should be prepared as described in the kit data booklet, and once prepared, they are ready for use in the manual assay procedure. All of the kit components except bottles 2 and 4 are used as supplied. The contents of bottle 2 are dissolved in 12 millilitres of distilled water. The bottle is capped and the contents are mixed thoroughly to ensure complete dissolution. To use bottle 5, the kit standard, dissolve 80 milligrams of its contents, acetaldehyde ammonia trimer, in 1 litre of distilled water and mix thoroughly. Follow the manual assay procedure as described in the data booklet. Pipette all assay components except the final trigger enzyme in bottle 3 into each assay tube. Acetaldehyde is highly volatile, so it is important that all assay tubes are capped after each reagent addition. A blank reaction and a standard reaction must be performed with each batch of samples. Pipette 2.0 millilitres of distilled water into all assay tubes. Pipette an additional 0.1 millilitres of distilled water into blank assay tube and cap all tubes. Pipette 0.1 millilitres of sample to sample assay tubes. Pipette 0.1 millilitres of prepared kit standard into standard assay tube. Pipette 0.2 millilitres of solution 1 into all assay tubes. Pipette 0.2 milliliters of solution 2 into all assay tubes. When all of the components have been added and tubes capped, mix the tube contents thoroughly and incubate the tubes in the 37 degrees Celsius heating block for approximately 2 minutes. After 2 minutes, record the first absorbance reading A1 at 340 nanometers for all of the assay tubes. In this demonstration, we are using the Megaquant Wave spectrophotometer set to read at 340 nanometers. Alternatively, a recording spectrophotometer with 1 cm path length cuvettes can be used. Swirl to mix the contents of bottle 3 prior to dispensing. After recording the A1 absorbance value, pipette 50 microliters of the trigger enzyme aldehyde dehydrogenase into all assay tubes. Mix the tubes thoroughly and incubate them at 37 degrees Celsius for 4 minutes to allow the reaction to go to completion. Record the absorbance reading A2 for all of the assay tubes. This absorbance reading completes the measurement of acetaldehyde. The absorbance readings A1 and A2 of the sample and the blank reactions are used to calculate the acetaldehyde concentrations in the original samples. When performing this test using the pre-installed protocol on the Megaquant Wave spectrophotometer, the results will be automatically calculated and printed via the onboard printer, or the data can be exported to a computer using the SF Capture software. Please see your Megaquant Wave video for further details. If the results output are raw absorbance values for both blank and samples, the calculations of acetaldehyde content can be performed manually as described in the calculation section of the kit booklet. Megazyme has also developed specific Excel-based MegaCalc applications for each Megazyme kit to allow quick and easy results analysis. Results can be analysed using the MegaCalc application specific to this acetaldehyde kit, which is available to download free of charge from the Megazyme website. The MegaCalc spreadsheet provides full instructions for use. Open the MegaCalc worksheet and input the following: sample details, absorbance readings for the blanks. For each sample, input the sample identifier and the absorbance values for the samples. Alter the sample volume if a volume other than the default 0.1 milliliters is used. If dilution of the sample has been performed, then input the dilution factor used. If no further dilution was performed, the dilution factor is 1. When all of the data has been entered, the concentration of acetaldehyde in the sample is automatically calculated and given as grams per litre in the original sample. For solid samples, input the concentration of the original sample extract in grams per litre. The concentration of acetaldehyde in the solid samples is then automatically calculated and provided as grams per 100 gram in the original sample.